Hello and welcome back to the Deep Dive. Today we're digging into a really powerful statistical method in genetics,
Yeah.
particularly useful in areas like, say, plant breeding. And our source material for this one is really fascinating. It comes from transcripts and class notes from Professor Guilherme de Silva Pereira's course. It's FIT 678 Genetic Data Analysis for Plant Breeding over the Department of Agronomy at the Federal University of Viçosa. That's pronounced Viçosa. So we're going to unpack something called Composite Interval Mapping, or CIM, and look at why it was, well, such a big deal for geneticists working on complex traits. Let's jump right in.
Okay. So the real challenge, when you're looking at traits like crop yield, maybe disease resistance,
Yeah.
these things are usually controlled by lots of genes, right? Not just one.
Multiple factors involved.
Exactly. And these genes, or QTLs, quantitative trait loci, they can often be physically close together, linked on the same chromosome. And the older methods, things like single marker analysis or even standard interval mapping, they really struggled with this linkage issue.
How so? What was the problem?
Well, they basically scan the genome looking for one QTL at a time. But if you had, say, 2 or 3 important genes all clustered together, the methods would often just see, like, one big blurry signal.
So you know something's going on in that area.
Yeah,
But you can't really tease apart the different contributions if they're close.
precisely.
Yeah.
You couldn't pinpoint the individual effects. And Professor Pereira's course notes, they really emphasize this in the plant breeding context, trying to find specific genes to improve crops when everything's tangled up genetically.
Okay, so that sets the stage. CIM comes along. What's the clever trick it uses to cut through that complexity?
Right. This is where CIM gets smart. The key innovation, really, is that it doesn't just look at the markers right next to the spot you're testing. It brings in information from other markers, sometimes spread across the genome, and it uses these as cofactors or covariates.
Cofactors, like statistical controls.
Exactly like statistical controls. Think of it like this. If you're studying, say, how genes affect human height, you might statistically adjust for sex, right? Because males and females have different average heights.
You control for that known variable to see the genetics more clearly.
Yes, CIM does something similar, but often uses other genetic markers as those controls.
Okay, so it's using genetics to control for genetics.
In a way, yes, and this is really powerful, especially in those plant breeding examples from the Viçosa course material. You might use general things like population structure in plants as a covariate, but CIM specifically selects markers, maybe markers known to be linked to the trait elsewhere, or even on the same chromosome, but, further away from the spot you're currently testing. The statistics let you test for a QTL in one specific interval while accounting for the effects picked up by those other cofactor markers.
So it sort of isolates the signal in that little window you're looking at.
It helps isolate it, yeah. It subtracts out the noise or the influence from those other linked QTLs captured by the cofactors.
That sounds like it would make a huge difference. So how does the actual analysis work then? You're still scanning along the chromosome.
You are, yeah. You move along interval by interval. But the statistical test at each point is different. It includes the potential effect of a QTL right there, plus the effects of all those chosen cofactor markers. You're asking, is there a significant effect here even after I account for what those other markers explain?
Okay.
And there's a neat detail, really emphasized in the course notes, defining a window. When you test an interval, you deliberately exclude any markers inside that immediate window from being used as cofactors.
Okay. And why is that?
Well, you don't want a marker that's super close to your potential QTL, maybe even part of the same effect, to act as a cofactor for it.
Because it would sort of soak up the effect you're trying to find.
Exactly, you would steal the signal. So you exclude the very close markers as cofactors to let the local effect stand out, while still controlling for the background using more distant ones.
Right, that makes sense. So given all this, what were the main wins for CIM compared to the earlier approaches?
The big advantages were, first, significantly higher statistical power. You're just more likely to find a real QTL if it exists. And second, much better resolution. So the ability to separate those linked QTLs,
Okay.
where interval mapping might show one broad hump, CIM could often resolve it into, say, 2 distinct peaks. Much clearer picture.
And for plant breeders, that means getting closer to the actual genes they want to work with.
Absolutely crucial for marker-assisted selection and targeted breeding.
It definitely sounds powerful, but no method's perfect, right? What are the challenges or maybe the downsides?
It's true. It's more complex. The key thing is choosing those cofactors.
Yeah.
Which markers do you include? The best set kind of depends on where the real QTLs are, which you don't know beforehand, so it can be a bit tricky. There are statistical methods to help select them, but it's an extra step. Also, if your markers aren't evenly spread out across the genome, that can affect the analysis. And while it's great for straightforward additive effects handling really complex gene interactions like epistasis within the standard CIM framework, that's not always simple.
Right. So pulling this together, composite interval mapping, CIM, was this really significant advance. It used these covariates and cofactors, often other markers, maybe other traits too, to basically help untangle the mess of linked genes, giving researchers more power, better resolution, a clear view of complex traits, which is obviously vital for things like plant breeding, like we saw in the Visosa course material.
Yeah, it takes this really complicated problem and uses smart statistical modeling to make it, well, much more approachable. You can see things you might have missed entirely before.
What's really interesting to me is how this core idea using statistical controls to isolate an effect is so fundamental.
Definitely. Those cofactors let you sharpen your focus on the specific thing you're trying to measure, filtering out related noise.
And here's where it gets really interesting for you, the listener. Think about that strategy, controlling for confounding factors, whether they're linked genes or completely different variables in another complex system. It's such a powerful concept, isn't it? Applicable way beyond just genetics. So what really stands out to you about how adding these cofactors fundamentally changes the search for specific causes within any complex web of influences? Something to think about.